Thanks, Julie, also for the invitation to come and present our mouse data. I spoke at this conference a couple of years ago, and it was a fantastic opportunity to meet many of the parents involved, and also Julie and the rest of her team. And it's actually been an exciting couple of years since then. So what I'm going to do is give you an update on our studies, which really focus on mouse models of epilepsy. And we're starting to make some progress now into understanding how this particular very interesting form of epilepsy is caused at the cellular level. The way the neurons are formed within these mouse models is certainly different, and I'm going to give you some information about what we've discovered over the past couple of years. All right, so I'm not a clinician. You see, I don't wear a tie. I'm a scientist, but it really is great to be in this environment where I'm speaking to quite a broad range. So I've tried to pitch the talk so that we've got hopefully something for everyone in terms of what they can get out of this particular talk. All right, so this is really a redundant slide. Nicola gave a fantastic clinical overview of the condition. So I'll just briefly go through it. Of course, it mainly affects females. There's a couple of mosaic males now in the literature, but clearly it's a disorder that affects girls. And one of the points that I really want to underline is it's highly variable in terms of the symptoms. The seizure onset occurs obviously during infancy, principally febrile seizures. Clusters of seizures are clearly a feature of this form of epilepsy, and intellectual disability is only present within some of the girls as well. So there's a lot of phenotypic variability, a lot of variability in terms of the symptoms. And this can occur even within families. So even with individuals with the same genetic change, there can be a lot of variability across that particular family. And what I'm going to spend a little bit of time at the start of the talk is telling you why we think that variability occurs. So Protocoheron 19 epilepsy, as I'll refer to it for the rest of the talk, is actually a very, very unusual form of inheritance. And this probably isn't a point that we've had underlined so far today, but there are, I guess, two forms of X-linked inheritance, and the one I've shown up here on the left-hand side is the most traditional form, if you like, X-linked recessive inheritance. And this is a form of inheritance where affected individuals are males. We'll get this working. There we go. All right. Down somehow. It's reversed, I think. Anyway, squares indicate the males. Clearly the males are affected with this form of disorder of inheritance, and males are XY, one X chromosome, one Y chromosome, and with mutations of genes on the X chromosome with X-linked inheritance, there's males are affected because they only have one X chromosome, and there's no second backup copy, if you like, to provide protection against that particular genetic change. And haemophilia is a common example of that. So that's the traditional X-linked recessive form. On the other hand, Protocoheron 19, as you all know, is completely switched on its head in terms of the mode of inheritance. And I've said this is almost unique, or unique in inverted commas. There's only one other disorder, as far as I'm aware, that has this very unusual mode of inheritance. And when I speak to this at conferences and amongst colleagues, they really spend about 5 minutes or so just looking at me saying, this is really unusual. You mean that girls are affected? They're heterozygous, they've got two X chromosomes, the mutations on one of those X chromosomes, and yet boys are not affected, and they've got no X chromosome, they've got no backup X chromosome to protect them. So it's a very unusual mode of inheritance, and it's really sparked a lot of interest in our lab to try and understand what really is the cause of this particular unusual form of inheritance in epilepsy. Okay, so we now know the gene. Joseph, who's sitting in the audience, who will be speaking later today, published this paper, I guess, a number of years ago now, identifying Protocoheron 19 as the cause of this form of epilepsy. And we've seen a little bit of this already. The gene is shown here underneath. There are 6 parts to it. It's not really so important, except to say that this is a form, it encodes a protein which actually lives on the outside of the cell. So this part of the protein shown in red sits on the outside of the cell, and it's important for sticking cells together, as I'll tell you later on during the talk. There are now many, many mutations in Protocoheron 19 that have been identified, over 100 different mutations, I understand, from recent reviews, and this is a little bit out of date now. But really the message I want to get across to you from this particular slide is that, what we think anyway, is that most of these mutations, if not all of these mutations, are affecting the protein in such a way that it loses its normal function. So for example, many of these changes which you see across the top there, they're genetic change that genetic changes that will truncate the protein to make it shorter so it's not functional. It will sometimes be degraded within the cell at the message level, which means it will not be functional. Or in cases where the red lines are underneath here, these are complete deletions of the gene itself, so they're clearly loss of function. And I guess the two things that I want to focus on today are really to tell you that, or discuss the phenotypic variability in affected individuals. As I said, individuals with the same genetic changes can have quite variable symptoms. I want to discuss why that occurs. And also, for the second part of the talk, I'll tell you about why these genetic changes that we see here, how they affect brain development using mouse as a model system. Actually, there was a laser pointer that one of the other presenters had. That would be great if it was available, because I've quite a few things to point out in the slides in the second half. Okay, so to explain why we think it's such a variable condition, I need to sort of go back to Genetics 101. So forgive me if this is information that you know already, but I'm going to tell you a little bit about X inactivation. And this is a process that occurs in all mammals. It doesn't occur in the zebrafish, as we heard earlier, but it's a process whereby males and females need to compensate for the fact that they have different sex chromosomes. So females are XX, as you know, males are XY, and that means that the 1,000 or so genes on the X chromosome in females need to be compensated in some way, because there's twice the dosage in females versus males. So this needs to be corrected in some way, and the way that the cells during development correct for this is to undergo this process called X inactivation. So in females, one of the X chromosomes, and this is a random process, is inactivated or completely shut down. And this occurs during early development, and once it occurs within a particular cell, it's permanent within that cell. And it's a random process, okay? To put that in a more visual terms, here we've got an early embryo shown at the top of this slide with this circle, and the two different coloured lines represent the two different X chromosomes. A blue one, or let's say a black one, and a brown one. So what happens during development is the cells decide specifically to shut down either a brown chromosome or a black chromosome, and that will, there we go, and that'll give rise to two different types of cells which are going to be permanently, have this process permanently in them through the rest of their life and all progeny from that particular cell. So what actually it results in is a mosaic. So this is a female brain, if you like. It's a mosaic of two different types of cells, depending on whether they've undergone X inactivation in this cell or in this cell. And there's a very widely, I guess, well-known manifestation of this. As I said, it occurs in all mammals, and in fact it's responsible for the coat colour in these calico cats. So if you like, where the location of the black cells are is a random process, and it's really a visual readout of X inactivation. And as you can see, each cat is in fact different. These are two different female cats from the same litter. They've got quite different coat colours because of this process. So the point really here is that there's a lot of variability that occurs because of this X inactivation process. So we now consider that in the context of protocoherin 19. This is a gene that lives on the X chromosome. Okay? So that means that females have two copies of this gene, and one of those copies of this gene has a mutation in it, indicated by the star, and the other X chromosome has a wild type or a normal copy of this gene. So the importance of this is that due to X inactivation, it means that in females, the location and the number of cells that will express this mutated copy of protocoherin 19 will be different in every female. So every female brain is different because it is a unique mixture of wild type and mutant cells. And this means that potentially there is scope for variability of the symptoms associated with this particular form of epilepsy, because each female brain has a unique mixture of wild type and mutant cells. Okay, so we think this is very important in terms of driving this variability that we see across individuals with the same mutation. Okay, so how do these mutations in protocoherin 19 affect brain development? As I said, we've been really studying this for the last couple of years, a little bit longer in fact, and using mouse models as a way to get to, I guess, the mechanism that causes this form of epilepsy. So why do we use mouse models? Well, this again is a slide I think redundant for many of you in the audience, but let me just illustrate why we think this is a good model system to look at epilepsy and in fact other disorders as well. This is not an epilepsy syndrome, this is called Waardenburg syndrome, and it's due to a gene mutation called c-KIT. And what I really want to point out to you is this characteristic lack of pigmentation on the forehead here. This is one of the features of this particular syndrome. And it's known that if you make a mouse model that has a mutation in the equivalent mouse gene to what's mutated here in these patients with Waardenburg syndrome, you find a very, very similar phenotype that's indicated by this mouse here on the right-hand side. So this is, if you like, a textbook example of how useful mouse models can be to understand the pathology that underpins many genetic disorders, including epilepsies. And that's really what my lab was interested in, is to try and understand how the brain develops from a very simple epithelium that forms in early embryogenesis. And the way that we do that is we try and particularly understand how diseases that affect early brain development impact on the way the neurons form. And this is, of course, Protocoheron-19 is one of these diseases that we work on. And we do this by generating mouse models that have, for example, loss of function mutations in the same genes that cause these human disorders. And on the left-hand side, you can see a human embryo at around 6 weeks of development. And on the right-hand side is a mouse embryo at around 12.5 days of development. So even though the final structures of these two embryos, what they develop into, is quite different, they go through a period of development where the brain development in particular is very similar. And this allows us to model these forms of epilepsy. Okay, so a little bit more about coherence before I move on to our mouse data, just to really give you some important background information on how we think mutations in Protocoheron-19 affect the way the brain develops. So coherence, as it's already been alluded to, this is actually a large family of molecules. Protocoheron-19 is just one of many coherins that are in the genome. The name comes from calcium-dependent cell-cell adhesion molecules, which gives you a hint as to how they work. Basically, these are molecules that sit, as I said, on the surface of the cell. So here are two cells, one on the left-hand side here, another cell on the right-hand side here, and this is the part of the protein that sticks out from the cell. And this is important for sticking cells together. So the message here is that Protocoheron-19 is important for sticking cells together, and particularly other cells that express Protocoheron-19. So this is called homotypic cell-cell adhesion. So Protocoheron-19 belongs to a family of proteins of which there are over 70 members. This really isn't important to get too much into detail of this, but I just want to give you a sense that this is just one of many proteins that has this similar function that's expressed from within the developing brain, and it sits down here amongst the delta 2 family of proteins. So a little bit of functional data. This is not our work. This was published a few years ago now, just to show you that Protocoheron-19 does seem to act as a cell-cell adhesion molecule. So this is just a simple experiment where you just take a group of cells that don't have any protocoheron in them whatsoever. They're called L cells. You just culture them in the laboratory. And what they did in this experiment was just to add some Protocoheron-19 to these cells. And this changed these cells from a non-adherent phenotype to an adherent phenotype. So this is exactly what you would expect for a protocoheron. The other important point is that they now did a more complex experiment where they had two different types of cells. They had Protocoheron-19 positive cells, shown here in red, and a different protocoheron, Protocoheron-10, and they put them in the dish together and asked the question, what happens? Do they segregate into different populations? And indeed, that's exactly what they do. So Protocoheron-19 expressing cells aggregate, as shown here, but if you put them in an environment where they have a choice to mix and stick to cells that express a different protocoheron, they're not able to. They tend to segregate and just stick with their other protocoheron positive cells. This gives us a sense for what Protocoheron-19 is doing during brain development. Okay, so is Protocoheron-19 expressed during development of the mouse brain? And indeed it is, and we were able to show this a number of years ago in Joseph's original paper, and it's expressed in parts of the brain that you would expect to be important for epilepsy. It's expressed in the developing cortex, which you've heard a bit about, but also strongly within the developing hippocampus. Well, it's in the right place and time to cause epilepsy, and we think the mouse is a good model because it's expressed also in the right place and time to be associated with epilepsy. And this slide really has been cut off a little bit here. It's probably the most important panel on this slide. Don't need to take in the detail of this at all, but it's really to give you a sense of it's a lot more complicated than what I'm presenting to you and what I'm able to present to you in this time slot. There are an enormous number of these coherins and protocoherins, as I said, within the genome, and they all have very complex expression during brain development. And the idea is that they form a code so that each cell within the developing brain has a code of protocoherins, and that code is important for allowing the cells that also have the same code to stick together and associate during brain development. So if you start to mess with that code by taking away a copy of protocoherin 19, that's going to cause potentially cells not to associate that would normally associate during brain development. And that's essentially the message of the talk. Okay. So really, I'm not going to go through the details of this because we now know the answer. And this is a question that's fundamental to what my lab is interested in. What causes the unusual inheritance pattern of protocoherent epilepsy? There are a number of possibilities which have now been excluded, and what we've come to settle on is a model called the cellular interference model. And the cellular interference model, which I'll explain over the next few slides, is based on the fact that during development there's a mixture of wild-type cells and knockout cells, in other words, mutant cells, within the developing brain that causes this particular form of epilepsy. And this is how it works. So, if an individual is a wild-type individual, in other words, has no mutation in protocoherin 19, as I said, there's this complex code of protocoherin molecules that allow cells that are identical, if you like, in terms of that code to associate during development. If, however, you've got a mutation in protocoherin 19, but it's present in all of the cells, for example, the transmitting male, in this case, the code isn't really, it's only interrupted in terms of protocoherin 19, but the cells can still manage to associate with each other reasonably well because there are many other forms of protocoherins that allow that association to happen. Where we get into the cellular interference model is that there are two different classes of cells. So this is what happens in the brains of individuals that have protocoherin 19. It's hypothesized. There are two different types of cells due to X inactivation, those that lack protocoherin 19 and those that contain protocoherin 19. And because this is an unusual mixture, because of the mosaicism that's caused by this X inactivation pattern, this can give rise to epilepsy and intellectual disability because the cells don't associate in the way that they normally would during brain development. And we wanted to essentially model this, and we've chosen to use mice as our model species. Okay, so how do we do that? Well, in mice, the technology has advanced a lot, but we used a traditional form of the technology. In fact, we were able to acquire a mouse which has a mutation in protocoherin 19, which was alluded to earlier. This lives on the X chromosome in mice. So mice enable us to really look at this mosaic X inactivation aspect of protocoherin epilepsy in a very good way. Okay, so these mice, for the aficionados in the audience, they have a lac-Z reporter allele that's knocked into protocoherin 19, which simply means that these mice have a mutant form of protocoherin 19 that has no function but turns the cells blue that would normally express protocoherin 19. And the fact that it's got no function, we think is a good model because it is consistent with the mutations that we see in patients that are loss of function. We've confirmed that in fact there's no protocoherin 19 in these mice. So this is a Western blot just showing you with an antibody that the mice which are males that have an X chromosome with a mutation on it have no protocoherin 19. So that's good. This is confirming really that we've got a validated model to do our experiments. So we've started to look at this model, and this has been recently published, some of this work, and I'll show you that at the end. But really these mice are remarkably normal, and it took a long while before we could start to see a phenotype of these mice. So let me tell you briefly about the brain morphology, in other words, the way the brain looks in these animals, and it's not really very much to tell you because it actually looks very, very normal as far as we can tell. So here we can see, for example, this is a male animal, which we're looking at the cortex here, and we're looking at the hippocampus here, and the cells that normally express protocoherin 19 are shown here in blue, and we can see it's got a very nice expression in the cortex and the hippocampus, as we expected. Here's a heterozygous female. sorry, this is all hippocampus, sorry. We can see the expression here in the hippocampus, but what I wanted to point out was that we've got this mosaic expression happening here. So some of the cells are blue, but not all of the cells are blue, and this is due to mosaicism. As I said, X inactivation causing this random expression or not of predicate here in 19. So we see pretty well a normal looking hippocampus, we see a normal looking cortex, and we see a normal looking cerebellum in these mice. When we start to look more closely at the morphology of the brain, so these are just sections of brains that we've stained and used histology just to look to see whether the cells look to be in the right place or whether there's any gross features that are abnormal. We can't really see anything going on in either the heterozygous or the knockout mice. And we look more closely by making measurements and so on. But the take-home message here is that the brains in these mice look remarkably normal. We started to look at cell migration by looking at the location of the blue cells during development to see if we could see any phenotype there, and again, there was not much to be seen. So it was a little bit disappointing really, but a couple of years ago things started to get more interesting when we started to look not at the morphology of the brain, but the activity of the brains. So we're looking now at electrical activity within these mice. And I should say we haven't seen any evidence of frank seizures within these mice. We've had them for a number of years. But seeing Nicholas talk earlier, seizures can, the early onset of seizures is difficult for us to model in mice because at that stage of development they're really associated with their mothers and they haven't been weaned yet. And the offset of the seizures might make it quite difficult for us to see those in mice. So what we've done instead is we've taken mice that are early adults and looked at their brain activity to see if we could find any evidence of abnormal activity. So this is the experiment up here. It's an EKG reading and it's a video EKG reading. Okay. So we're looking at the cortex here. And even in normal mice, there are so-called spike wave discharges that occur within these animals. But this is where things got interesting. In heterozygous mice, we can see that the activity is, of course, much more elevated, and the frequency of these spike wave discharge events is also considerably elevated, as I'll show you on the next slide. And I guess even more satisfyingly, we've now done a few mice that are, you can't see it down here, but this says knockout. So these are mice that are females, but they have no predicate here in 19 function. This goes back to a more normal looking pattern when we look at the spike wave discharge pattern. So when we look at all these data over the extent and count the number of spike wave discharges per hour, this is a wild type spectrum here, and you can see there's some variation here as you'd expect in wild type mice. But the most interesting thing here is that the heterozygous mice, which model the disorder in humans, have this elevated spike wave discharge. So we've now finally been able to determine that these mice, the electrical activity in the brains of these mice is indeed affected, and it occurs in the heterozygous mice but not in the knockout mice. So this reflects exactly what we see in humans in terms of the mode of inheritance of the epilepsy.
What's the background, Paul?
Sorry?
The mouse background?
These are DBA. In fact, we crossed them onto DBA for 10 generations to increase the propensity for electrical activity. We haven't done it on the Black 6 version that we had originally. Okay, so this was good. This was encouraging. We've now got a phenotype in mice that we can identify that is abnormal within the brain. Okay, so the final question I'm going to tell you about is really to understand X inactivation. I'm expecting someone to come through there in a minute. Okay, so just to frame that question again, we've got this X inactivation event. So imagine early during development, there are 2 X chromosomes in every female cell within the brain, and there's this random event whereby one of the cells will express predicate heron from one of the X chromosomes, and one of the cells will express predicate heron from the other chromosome. And this occurs in all the cells so that you get this mosaic effect of cells that are positive or negative for predicate heron 19. So we wanted to look at the cellular interference model, and in order to do that we needed to identify which cells were predicate heron 19 positive and which were predicate heron 19 negative. Okay? We need that. We need a tool to be able to enable us to do that, to test this cellular interference. And what inhibited or we could not do these experiments for many is because we didn't have an antibody which enabled us to detect these wild-type cells within the brain. It's a common problem in biomedical research, and it's one that's really prevented a lot of progress in this, answering this particular question. So we used the CRISPR technology that Anna nicely described earlier to modify the endogenous gene so that it would now be able to be detected by an antibody, which we could buy off the shelf. So essentially what I'm telling you here is that we've modified PCDH19 gene in a mouse in such a way that we can now detect the protein, the wild-type protein that it produces. So this got us around this technical problem that we had. And we did various validations of that. For those of you interested, we put a flag and an HA tag at the C-terminus of PCDH19, and it works very nicely by Western blot. And also, we can now be able to detect in mouse brains, for the first time, we've been able to detect the location of the PCDH19 protein. And this is a control on the right-hand side that lacks this tag. It's a negative control, which we're not expecting to have any signal, which is fine, it doesn't have a signal. But on the left-hand side, you can see, shown in red, all of the cells in the developing mouse brain that express PCDH19. And it's actually quite a large number of cells, but I'd like to point out a couple of different types of cells that are important. There's a group of cells here in the ganglionic eminence, and these are cells that are fated to be a form of neuron called inhibitory neurons. And these cells migrate in this direction initially, and then they migrate tangentially across the top of the cortex before dropping into the cortex themselves, and they're responsible for limiting the amount of excitatory activity of cells within the cortex. So that's one interesting population of cells. The other interesting population of cells are these cells across here, which are precursors for excitatory neurons in the cortex. So quite an interesting spectrum of cell types that this particular protein is expressed in. And we've done some more labelling studies again for those of you who are interested, defining exactly. It seems that PCDH19 is expressed probably in all of the inhibitory neuron precursors, and it's certainly expressed in amongst, we think, all of the radial glia cells within the developing cortex. Okay, so quite wide expression within these regions. After birth, expression persists. So this is a mouse brain at 7 days after birth. And you can see this continues to be quite strong expression within the cortex. And you can see it's just restricted really to one region of the cortex, which we've now defined as layer 5A for those of you who are interested. So PCDH19 expression persists in layer 5A within the cortex after birth. Okay, so we had a handle on where it was expressed, and now we wanted to test the cellular interference model. This is a little bit complicated, so I'll try and explain it as simply as I can. But what this experiment involves is crossing the mice that have this tagged form of PCDH19, which we can use to detect wild-type cells, and crossing them with the knockout model, the loss-of-function model, which I explained earlier, which has cells which turn blue when they lack PCDH19. And by crossing these mice together, we can come up with a number of different scenarios, controls and test, and I'll just explain what they are now. The controls, which I won't spend a lot of time showing to you, effectively that's what I've shown you on the last couple of slides. All the cells are wild type and they're red and they contain wild type PCDH19. We can also make completely knockout mice, which I showed you with the blue cells earlier, and these mouse brains appear to be normal, and in this case all of the cells that express PCDH19 are shown. And now, I was showing you in blue, but now I'm going to show them to you in green. The really interesting models, though, that I'm going to tell you about now, the next couple of slides, are these two. This one is a heterozygous female, so this represents the same genetic changes that we see in girls with PCDH19 epilepsy. And what we're looking to test here is when we've got wild-type cells, shown in red, and we mix them in the brain with cells that lack PCDH19, which will be shown in green, do we get the segregation of the two types? That's really the test. That's the cellular interference model. On the right-hand side is a control for this experiment where we've got cells that are wild-type for PCDH19, shown in red, with other cells that are also wild-type for PCDH19. This is just a control to see what the brains look like with just random X inactivation. Okay. First of all, let's look at the control. control brains look like this. It looks a lot like these are just wild type brains, and there's a little bit of X inactivation. As you can see, we see slight patches here. Nothing really terribly remarkable in terms of the expression. But now when we do the experiment to look at the cellular interference model, we see quite a striking difference in terms of the pattern of the cells within the developing brain. So we can see here in the developing cortex, we've got these patches that almost look like tiger stripes, and what they are showing you are cells which express protocoherent 19 segregating away from cells that don't express protocoherent 19. In other words, wild type and mutant cells are repelled from each other during brain development, as the cellular interference model would predict. Now we're just looking at the mutant cells in the same genotype. So this is just showing you, if you like, the inverse of the red pattern that I showed you on the last slide. And when you put these two together, this is what we see, this rather beautiful pattern where we've segregated away the green cells, which are mutant cells, from the red cells, which are wild type cells. Now, what impact this has on brain function to cause those EEG phenotypes, which I showed to you earlier, we're still trying to discover. But for the first time, we've really been able to do two things. We've been able to show that the mice have electrical differences in their brain, and that the way the brain develops in these individuals that reflect the same genotype as protocoherent epilepsy is quite different. There's a segregation of wild type and mutant cells. The other point I'd like to make is that, before I go on to that, we've actually also nailed the time point at which this starts to occur during brain development. So you don't really need to know the details of this, but 9.5 is just the time, the embryonic day, which we first see protocoherent 19 expression starting to appear. And at this time point, really this segregation phenotype hasn't properly kicked in yet, because the cells are still mixing with each other and trying to work out whether their partners are positive or negative. But by 10.5 days of development, we see quite clearly in these cortical progenitor cells, so these are radial glial cells, for those of you who know this area, are already segregated in terms of positive and negative cells. So quite an early phenotype during brain development. And the last point I really want to make is that this effect is variable, highly variable. So what I've shown here are a number of different animals, or different embryos if you like, brain sections from each of those embryos. And the point that I want to make here is that each of these has a unique pattern of positive and negative cells. So even though the genotype, in other words, that they're all heterozygous for the same mutations, even though they're all heterozygous for the same mutation, we see that the output of this during brain development is quite different. The pattern of X inactivation means that we've got different patterns of positive and negative cells. And as I said, we think this is really important in terms of the variability that we see in patients with this particular form of epilepsy. And just coming back to that, again, you can't see it on the right-hand side, but I've shown you this before. You can see that we've got a spectrum of phenotypes in terms of the spike wave discharges, and some of them actually go back into the normal range shown here on the left-hand side. The point I want to make is that it's quite a wide spectrum of spike wave discharge frequencies in the heterozygous mice, and we can see also there's quite a variety in terms of the pattern of the wild type mutant cells that we see during brain development. And we think these two things are linked. What we now want to understand is how these two observations are linked together. Okay, so just to finish off, really, again, for those of you who are interested in this area in terms of what's really happening at the cellular and the molecular level on the surface of these neurons, that's really where we're taking some of our research into the future. So as I've told you, protocoherent 19 is implicated in this homotypic cell-cell interaction, and I've shown you the phenotype of the heterozygous mice. We also know that neurons express many other protocoherent 19s, so why don't they sort of step in and compensate for the fact that we've just lost one protocoherent 19 in these cells during development. So we want to look more closely at this issue and ask the question, what's the mechanism that underpins this particular cellular phenotype? And there's actually been a lot of work in this area in the last year or so, and it's come out of structural studies by Barry Honig, Tom Maniatis, and others over on the East Coast. And there's quite a nice model now to describe the shape of these protocoherent molecules, and also how they interact with each other at the cell surface. And this is the kind of model that's been developed. It's sort of the take-home message here is that there's like a lattice work of these protocoherin 19. So it's not as simple as just one meeting one other molecule, but rather they get together to form this lattice work. And when this lattice work is a perfect match between cells, then it sticks the cells together. But as soon as you make a single mismatch in this lattice work, for example, take away protocoherin 19, it has a massive impact on the ability of these two cells to associate. And some of the structural studies that govern this have been very informative because they've indicated there are so-called trans interactions that occur between cells, but also cis interactions that occur on the cell surface. And we've started to probe this in a little bit more detail. This has already really been confirmed by studies in cells that this occurs with protocoherin 19, but no one's shown to date that protocoherin 19 cells can interact with other protocoherins on the surface of the cell. So we took on that as a project in the lab, and a fine PhD student who I'll introduce to you, at least on the board at the end of the talk, has been looking very carefully at this. And the take-home message from this is that protocoherin 19 can indeed form cis interactions on the cell surface. This is an IP plot showing that it will form interactions with itself, with protocoherin 17 and 10, so other members of the same family. And what we think now is that this model is probably a pretty reasonable estimate of what's going on during brain development. So we've now got a structural model that we can probe further to understand how the cellular defect is occurring. Okay, so in summary then, what I've told you really is that protocoherin 19 mice have increased brain activity, and that's really been, I guess, one of the key findings that we've had over the last couple of years. And that the pattern that we see in heterozygous versus knockout mice is consistent with the inheritance pattern that we see in humans. We've been able to look at where the protein is expressed during brain development in mice. And as I've told you, it's in the excitatory neurons and the precursors, and as well as the inhibitory neuron precursors as well. And afterbirth is predominantly located in layer 5A within the cortex. The mosaic expression of protocoherin 19 leads to this striking cellular segregation phenotype in the cortex. So we think that cellular interference model that was proposed a number of years ago is correct. And we can conclude now that protocoherin 19 plays a critical role in maintaining this homotypic cell-cell adhesion interactions during brain development. And this is really the first in vivo evidence of the cellular interference model. And it gives us, I think, a real insight into what is occurring during development in brains of girls that have protocoherin 19 mutations. So I've got to finish with a model. Scientists like to finish their presentations with a model. So this is basically bringing it all together in terms of what we think is going on during brain development. So in a wild type, or if you like, a normal situation where there's no mutations in protocoherin 19, the cells get together during early embryogenesis, and at a point at around, at least in mice, around 9.5 days of development, they begin to express protocoherin 19. The pattern of X inactivation, though, occurs even earlier than that during embryogenesis. Now, in a wild type situation, it doesn't really matter because the X inactivation pattern doesn't really matter because all of the cells express protocoherin 19, whether they've been activated one X chromosome or the other, it doesn't matter. And this gives rise to a mosaic brain, but the cellular interactions are not affected. What we've got in the case of protocoherin 19 heterozygous mutations is something quite different. We've got the X inactivation occurring early. Once the cells start to begin to express protocoherin 19, they recognise that there's two different populations within the developing brain, and they segregate away from each other so that ultimately you get zones that have either mutant expression or wild type expression. And we think this is somehow causing those abnormalities in brain behaviour that we see elevated spike wave discharge activity in heterozygous female mice. And our challenge really is to understand the connection between those two observations, as I said. And the last point is that because every female brain is going to be different in terms of this X inactivation pattern, this variability drives different symptoms in patients that have the same mutations. So we published some of this stuff, and rather timely, it came out just a couple of days ago. So that was really wonderful to get some of the data out. And thanks also to Joseph and his group who have contributed to this paper as well. We would also very keen to acknowledge Protocoherin Alliance for their support in making this paper happen. So thank you again for that. And lastly, to introduce you to Daniel, who's a wonderful PhD or graduate student. And I got him to, perhaps slightly reluctantly, pose for this selfie photograph with his Protocoherin 19 Alliance shirt that I bought for him back from the meeting last time. He's done a wonderful job on this project with some assistance from Sandy and James. Joseph's a long-term collaborator on this project, as I said, you'll hear from him later. Also acknowledge Steve Petcher and Kay Richards for the EKG readings on the mice, and thanks to Freddie here in 19 Alliance and the Australian Government NHMRC for support. And with that, I'll close and take any questions.
Beautiful talk, Paul. So, two questions. One, did you see with your knock-in PCDH19 expression in the parvalbumin basket cells? And the other question is, in your clusters, what's the makeup of excitatory versus inhibitory neurons?
Yeah, both good questions. The first one I can't tell you because we're still doing that study. The second one, again, it's really a work in progress. I probably didn't show this, but after birth, as I said, the layer, say 7 days after birth, the layering that we see that occurs during embryogenesis is maintained. We see expression in layer 5A, predicate neuron 19, but only in, if you like, in columns within that heterozygous female. So in other words, whatever's set up during embryogenesis persists after birth. What we haven't got to the bottom of yet is, after birth, is expression maintained in excitatory neurons, or is it maintained in inhibitory neurons that are in that region, or in both? And that's a question that we want to get to the bottom of.
Yeah, I mean, I basically have the same question. I mean, it was a wonderful talk, really, really marvelous, and I feel like you are the brother I never had. Yeah, I feel that way, and well, you will see why. But anyway, do you think that this mechanism would be present at the time of synaptogenesis and at the peak, for example, the critical period for cortices?
Yeah. So I didn't go into it. Clearly it occurs before synaptogenesis. As I said, 9.5, 10.5. So really they're still radial glia. However, we know the expression persists later on during development, including to the time when synaptogenesis is occurring. It's certainly part of our future studies to look at that exact question, because we know that expression's there, so clearly there's scope for positive and negative cells to not form synapses where they should. And we're wondering whether lateral connectivity within the cortex is affected by the fact that you've got zones of positive and negative cells that are there. So that's certainly a question we're looking at. I can't give you an answer, but I think there's certainly scope for that kind of mechanism.
If I can add one comment, those zones are really large. I mean, for example, in somatosensory cortex, they would be basically larger than a single barrel. In visual cortex, I mean, 2 of those areas would cover basically the entire V1 area. So if there are preferential connectivity within or exclusion of connectivity, function is obviously going to be affected in a massive, massive way. And that, of course, would be extremely interesting to study.
Yeah, I agree. But it also makes it difficult to study because each mouse brain that we look at is different. So that's part of the issue with this project, is that it's been difficult to consistently find those phenotypes. When we started out, we needed to convince ourselves that it was real. Quite clearly it is real, but it took a long time and many numbers to convince ourselves that it was really real, because of that variability that you get with X Inactivation.
That's the last question.
Yeah, I have a technical question. So, there's a lot of red signal also outside of the cortex. Okay, is that a positive signal? I mean, is the expression of the protein also?
Do you mean in the postnatal stages or the early stages?
Well, I guess the few last images you
okay,
showed.
So it's in the marginal zone with migration of the interneurons. So that signal that you see on the outside of the cortex is migrating interneurons as they work their way around tangentially before they drop down into the cortex.
Okay, but in that case, there is no green signal outside. I mean, the mosaic is only present in the cortex, not outside. How do you explain that?
I'm sorry, I didn't catch that.
I mean, the green is only present on the cortex. It's green signal, so cell
Oh,
not
yeah.
expressed. So why is not?
Yeah. There's also another aspect to it, is that the localization of the laxed protein is actually perinuclear, whereas the protein 19 tag elaborates across the entire cell surface. So you actually see a different pattern when you look at the laxed stain versus the protein 19, because of where the protein is within the cell. Perhaps that's, maybe we can talk about this afterwards if that's.
Yeah, that's curiosity. If you grow this culture, mixed culture, grow this neuron in culture, not in vivo, so do you also this segregation or other?
We're doing that at the moment.
Christoph, do you want to ask the question?
Sure. A very quick question. So is it truly a dispensable protein, and are there any other protocadherin models or absent, like other absent protocadherins or loss of function other protocadherins that didn't show the same? Meaning, do the mice that are complete knockouts have any differences outside of the ECOG recordings that you have?
We haven't seen anything unusual about the knockout mice. And their ECOG readings, as you saw, are back down to normal. We need more numbers for that, but it's looking like they're in the normal range. Is it a dispensable protein? I guess under those definitions, then you would say yes. However, as you would know, in humans, there's some hint that's been in the literature about whether it's truly dispensable, and perhaps there's a subtle phenotype in transmitting males. I don't have any experience with that, so I can't comment on that further. What I would say, though, is that clearly this is driven by the mosaicism, at least that's our view. So we haven't done this experiment, but we thought about it, actually making mosaics of other protocadherin molecules that are present within the brain. We could do that with CRISPR, and that would be a very interesting experiment to see whether protocadherin 17 or 10 has a similar kind of impact if it were mosaic. So I would say, in a sense, it is dispensable, but because it's here in a mosaic form, that's what's driving it.
Thanks, Bob.